Hi everyone, welcome back to my channel. My name is Chacha, and today I'm going to be going over neurotransmitters. I previously in a different video went over hormones, a little bit of neurotransmitters, but I realized that the video was getting close to an hour, so I cut it and I was going to talk about neurotransmitters all by itself in this video. Once again, I am a student, so there may be mistakes in my videos, so feel free to call me out in the comments, and hopefully that this could be helpful to someone. And I'm making this video more or less to hold myself accountable while I study for the MCAT. With that out of the way, I took this from my prior video, but it's good just to go over like the difference between neurotransmitters and hormones. Um, so hormones are typically produced by the endocrine glands. So it's like your adrenal gland, like including like your adrenal medulla, adrenal cortex. How you could kind of like remember the difference between the medulla and cortex. So the cortex, its acronym is AC, and the hormones that it produces are aldosterone, androgens, which is the A, those are the two A's, and it's also cortisol. So it's in the name. Then the medulla will be kind of everything else. So that'll be like your norepinephrine, epinephrine, etc. Then your hypothalamus, which I like to think of sort of like the mob boss of your brain. So it's kind of like what keeps everything in your checks and balances, make sure that everything is in homeostasis. So make sure that your body is properly balanced and isn't going to explode or die. So very important. And then your ovaries and testicles, your gonads, sort of, and then your thyroid. Those are kind of the main ones, I feel. Whereas neurotransmitters are produced, hence the neuro, by neurons typically. So they're created in the soma, which the soma is like a fancy word for saying like the cell body, and they're stored in vesicles, in the axon terminal. So like right before, so it's at presynaptic, like if you're looking at a synapse, your axon terminal, this is a dendrite, and this is your axon terminal. And I will go more into this a little bit. So, yeah, they're stored in vesicles in the axon terminal. And in order for those vesicles to be released into the synapse, you need to have a depolarization event on calcium that influxes into the axon terminal. And then sometimes hormones can be released by neurons too. It's not like specific for endocrine, but just like a general rule of thumb, it is the endocrine gland. More specifically, like norepinephrine, that can be something that could be produced by neurons. Hormones tend to be much slower than neurotransmitters, and as a result, they tend to last longer. So this is a typical trade-off that you'll see a lot of the times in the MCAT. If something is slower, then it typically lasts longer. But then you have, if you need it like immediately, like if it's like a life or death situation, then you're then it's slow, so you're not gonna able to have it long. And then whereas neurotransmitters are longer. So that's like the immediate thing. So it's like if you're in a life or death situation, then you can have easy access to that neurotransmitter. But the drawback is that it doesn't last long, so it's not really a permanent effect. Then hormones act typically on like far away structures. Not necessarily outside of the brain, since like the oh my gosh, hypothalamus, that's like the main one, and then that'll go to your anterior, posterior, it's sorry, your anterior pituitary gland through that hypophyseal portal system, and I kind of go more of that in my hormone video. Whereas the neurotransmitters, they typically act on nearby structures like other neurons. So that one I was talking about, that postsynaptic and presynaptic cell, in that synapse. And then both are chemical messengers which bind to a receptor and will have some changes to whatever they're acting on. So typically there will be a signaling cascade and it'll have your body react accordingly. So what are some of the key players of neurotransmitters? So I wanted to more or less talk about like specifically in the synapse. So that little place in between the presynaptic, which I have in light blue, and postsynaptic, which is in that dark green. More or less, like how does neurotransmitters, how do they like get out of the synapse after they're in there in the first place? So first, let's realize how do they get released into the synapse to begin with. So in my axon terminal that I have here in light blue, I have so just pretend that there was an action potential that made its way throughout the neuron into the axon terminal. So then it's depolarized, right? Since it's like, let's just say it's like in that depolarization phase, that means that the voltage gated calcium channels, they'll get open because that axon terminal is depolarized. So it's positively charged. So calcium is going to get open, calcium is going to influx into the axon terminal, and then calcium over here, which I have in that little light green, Ca2 plus, it'll bind to a tiny little receptor on the vesicle that holds a neurotransmitter in it. Then it'll react in a signaling cascade. And that signaling cascade, it'll go, 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 and then it'll bind to the membrane of the axon terminal, and then it'll get exocytosed into the synapse. And then exocytosis versus endocytosis, which is like something entering the cell, is that exo, exit, endo, enter. That's how I like to think about it. And then the neurotransmitter, once it's in the synapse, it'll make its way, just like diffusing over to the receptor that it can bind to. And once it's bound to that receptor, it can either react in a depolarization event, or it can be a hyperpolarization event, which we'll see when we get into the more specifics of the different neurotransmitters. So now that we know how neurotransmitters get into the synapse, how do they get rid of excess neurotransmitters? So the first thing is that it can reuptake via transporter, which is what I was talking about, the endocytosis, so entering the cell. So it'll get taken back up into the presynaptic neuron, which I have in the light blue. So the presynaptic neuron is synonymous with the axon terminal, and then it'll get stored back into a vesicle. So it's just like that whole process sort of in reverse. Um, the other one is ACEs. So whenever you see something that ends in an ACE, when you're talking about like synapse, typically that means that it's going to degrade the neurotransmitter. So an example of this is an acetylcholinesterase, which is it gets rid of ACH, and we'll talk about what ACH does in a little bit here. And you know that it's going to be degradation for neurotransmitters due to that ACE that it ends in. And then diffusion is the last one. So this is sort of rare. Typically, this isn't what happens. The most common one is that ACE. In diffusion, typically like gases do it, but also other neurotransmitters can do as well that are in the gaseous form, like nitric oxide, not to be confused with nitrous oxide, which is not great for you. Sorry, Kanye. And then agonists, sorry, we should talk about the two main types of neurotransmitters. Like these are kind of like their classes that they can be in, if you will. So there's agonists, which are what mimics the neurotransmitter, which is like typically what a lot of drugs do. And we'll get down to here of like what an agonist can do. And then there's antagonists. So I kind of think of like antagonists are kind of like villains, and they're trying to stop our superheroes, which are neurotransmitters, from changing the world, or in this case, changing that postsynaptic neuron. So down here, I kind of have something that's related more or less to addiction. So like let's say you're taking an agonist for something. I guess like let's say at the beginning, like GABA and glutamate, those are, and we'll get more into like what they are in the beginning. But GABA is more of an inhibitory neuron, not neuron, neurotransmitter, whereas glutamate is an excitatory neurotransmitter. So they're in perfectly balanced, as all things should be. I look at that little fulcrum, perfectly balanced. Whereas let's say you take a substance that's an agonist for GABA. So as a result, you're in order to have that the fulcrum be like perfectly balanced, your body's just gonna up your glutamate amount so that it can function properly. Just because both GABA and glutamate are really important. Just because something is inhibitory and something is excitatory doesn't mean that you should have less of one versus the other. So in this case, because you're taking that substance that has GABA, then its glutamate is also going to increase. But let's say that so it's like you're increasing like kind of your tolerance. So like the more that you need to take in order to get that high, let's say for example, that means that you need to increase the GABA amount in order to have that same high. So that's kind of like your tolerance. And then let's say all of a sudden you're just like stopping like GABA, like taking your GABA agonist just like cold turkey. So as a result, your body has like your GABA levels will go back to normal just because you have the same amount of receptors, but because you're taking something that mimicked GABA's properties, it's still like you're still producing the same amount of GABA that you were producing before you were taking that agonist. Whereas for glutamate, because you weren't taking anything that was an agonist for it, and glutamate just had to increase its amount just in order to keep up with that GABA agonist, then your body's just like completely thrown out of whack. So you have an disproportionately high amount of glutamate and a low amount of GABA. So you have excitatory, so neurotransmitters going on versus there's not anything that can block it out. There's no balance left. So that's really bad. That's more or less kind of like what addiction is you're just like once you reach like a certain tolerance, like this is like more prone to happen. Or if you like attempt to quit a substance just cold turkey, that's why it's so dangerous, just because your body is just like oh thrown out of whack and it takes a while for like glutamate, let's say for example, um for this example, glutamate to like come back down to its regular levels so that these can be balanced. But it'll take a while for GABA also to increase too, just because it's so far behind, right? Like it's starting like if you're like a runner, right? Like if you're racing like let's say like glutamate's like Usain Bolt and Usain Bolt had like 100-meter head start from you and you're trying to reach the finish line. Like it was an impossible feat. Enough about that though. Let's kind of talk about the main neurotransmitters that you need to know for the MCAT. So there's GABA, which is like the inhibitory neurotransmitter, like the Ohio State, the GABA. So how does it work? So GABA, you don't really hear about this as much. Like chlorine, when you're talking about like what's inside and outside of your neuron, outside you have sodium, you're always talking about like sodium, and then potassium is inside the neuron. Whereas also outside you have chlorine. So you have anions outside that are in high concentrations, whereas inside you typically have various anions. They're not really super specific or what you need to know for them, Cat. So what does this do? So GABA will bind to the your neurons and it'll bind to the ionotropic channels. Also, occasionally, it'll bind to potassium channels too. So what happens? So once that GABA it binds to the receptor, it'll have chlorine because remember we were saying that there is a high concentration of chlorine outside the cell. So that means that chlorine will go into your neuron so that it'll be like super hyperpolarized. And it's less likely to have an action potential. Remember, you have that axon hillock, which is kind of like your accountant. It's like tallying up, oh, like you need like X amount of like voltage in order to have an action potential fire throughout the rest of your neuron. If you have GABA with your chlorine, then it's not likely to fire because you're adding uh, negative voltage. Remember, your resting membrane potential is around negative 70 millivolts, whereas in order to reach an action potential in your axon hillock, what it needs to be is negative 55. So if you're adding more like anions into your neuron, then it's less likely to fire. So now once you added, let's say if that chlorine is now at negative 70, negative 90, sorry. So not, it's even further away from that starting line, from that threshold. And then for like secondary GABA, sorry, I should say GABA when it does the chlorine, that's like the first like primary like receptors. Whereas the secondary receptors, it's equally as hyperpolarizing. It's when you have potassium because potassium, it'll be a potassium ionotropic channel. And potassium, remember, is at high concentrations inside the cell. So remember, it always goes from low to high. So once you have that channel open, like potassium is going to flow out. So that's another way that you could hyperpolarize because you're taking something that's a cation and transferring it outside of the neuron. So that's making it even more negative too. And how I kind of like to remember that GABA is inhibitory is GABA was a bad boy, so he's in timeout. Sort of more or less a thing. So timeout, more or less like hyperpolarization. So that means that action potential won't fire and your cells can't communicate. So no neurotransmitters released inside synapse. However, this is like kind of an important note, less likely to kind of just show up. But in early developmental stages, GABA was excitatory due to there being kind of like chlorine was increased like outside the cell versus I mean not outside, inside the cell versus I don't know why I said outside. Inside the cell. So once it bound to that receptor, then it would go outside instead, which would make it depolarized since you're having chlorine go outside. But that's something that's not really important. Um, then we have glutamates. So glutamate is the brain's primary excitatory neurotransmitter, except for in your eyes. That's a really important distinction to make. So when you're exposed to glutamate, it decreases like it's not like firing, like the neurotransmitter isn't going into synapse when you're exposed to lights. And then glutamate, it's what binds to AMPA and NMDA. Those are the receptors that it binds to. And instead of having that hyperpolarization like GABA, it releases depolarization. And I'll get more into what AMPA and NMDA are. So there's something that you need to remember. So there's like the Hebbian rule. So neurons that fire together wire together, which is something that you probably heard before, but just and then we'll kind of figure out why. So what is AMPA and what is NMDA? What type of, I guess, ions why is it important? So AMPA is an ionotropic glutamate receptor, which we were talking about, and it controls sodium channels, and it produces an excitatory signal, which can activate NMDA. So AMPA is really important because you need to have that AMPA activated in order for that NMDA receptor to have calcium go through to be activated as well. So there needs to be a depolarization event. And NMDA, it's blocked by magnesium. So like the two requirements that NMDA needs in order for calcium to flow through is that it needs glutamate to bind to it, and it needs that depolarization event from that AMPA receptor. So it needs an excitatory signal. In NMDA, it's because it's blocked by magnesium, it needs to be dislodged from the receptor, and it needs that excitatory signal to open that triggers a signaling cascade, and it allows for calcium to flow through. And it changes the proteins in the synapse and some structural changes. So once that calcium, once it enters into the postsynaptic neuron, then it can go and it can sort of access a transcript, not sort of, it accesses like transcription factors and it'll increase the amount of AMPA receptors that are on the dendrites. So overall, that means that you'll need less of the glutamate, less of that neurotransmitter into the synapse in order to have the same response, which is what long-term potentiation is. So that means that it's just an easier overall in order to reach that action potential. And then we could go into dopamine, which is associated with movement, rewards, motivation, attention, pleasure, and pain. So dopamine is primarily produced in the midbrain. So you have the ventral tegmental area, which is the VTA, which is involved in reward, motivation, and pleasure. I don't really have like a good acronym for that, or like not acronym, like kind of to remember. There's more or less like I went into substantia nigra, which I do more or less have like something to help remember me. But that's just how I'm like, if it's not the substantia nigra, then it's a VTA. But the substantia nigra, it's associated with movement. So that's what releases into the striatum. And the striatum is heavily associated with addiction. So like that's kind of what we're talking about here. But more or less addiction in that it's like more like pleasurable too. It's like, yeah, that pleasure more or less. And then also the basal ganglia, which is involved in movement and also implicit memory. Specifically, the basal ganglia is really important for procedural memory, which makes sense if you were to think about it, since dopamine is associated with movement, especially with the substantia nigra. Um, because like when it's procedural memory is like more or less, excuse me, riding a bike and stuff like that, where it's just like unconscious movements. That's how I like to think about it, and it makes sense. So it's the basal ganglia. Yeah, substantia nigra, striatum. That's just more or less how I think about it. They both have the S's. And then Parkinson's, so there's two hypotheses that are associated with dopamine. So for Parkinson's, it means that like if you have too little dopamine, um, so it's like when you have loss of dopamine neurons in the basal ganglia, which makes sense because we're talking about the basal ganglia is really in charge of like that movement and those unconscious memories, like movement memories, like the skills that you need, like riding a bike. So that'll also result in tremors too, just because like you don't have like control over your body anymore. And it just progresses and it gets worse and worse, the less dopamine neurons that you have. Because you have a limited amount of neurons inside of your brain. And if your brain realizes, hey, like there's not really a lot of like neurotransmitters that are being released onto this postsynaptic neuron, like the synapse is kind of like dead, then it'll actually prune said synapse. So that's a really big problem. Um, Especially for like those movement neurons, just because there's not a lot of action happening. So the brain doesn't want to waste, quote unquote, waste, I should say, resources. And then schizophrenia, that's when you have too much dopamine. So that's when you have like the positive symptoms of schizophrenia. And a lot of the times when you're thinking about like positive versus negative, positive typically means that you're adding something that wasn't there, whereas negative is when you're taking away something that wasn't there. So like positive would be something like hallucinations. So like if you have like auditory hallucinations, where it's like you're hearing voices that aren't there, or and also visual too. But typically with schizophrenia, you either have one of the sensory hallucinations, not the other. And visual isn't as common as like an auditory hallucination. Whereas negatives would be more or less like that catatonic state. So it's like loss of emotions. Uh, so then we can go into serotonin. This is one of the receptors that you actually kind of really need to know. So if you see like something like 5-HT receptor, then that's serotonin. And a 5 kind of looks like an S a little bit. So that's how I would remember like that would be for serotonin. Serotonin is associated with sleep, mood, eating, and dreaming. There's typically like if you have like too much meat, it's considered manic. This is like two of the hypotheses associated with serotonin, whereas too little is depression and or anxiety. So a common drug that you'll see for serotonin is SSRIs. So that's a serotonin like reuptake inhibitors. So what that means, like going back, yeah, to this is remember like the reuptake, that's when it endocytosis. So that's how the neurotransmitter will get back into the vesicles into the axon terminal or the presynaptic neuron. This basically prevents it from being endocytosed. So there's just like a ton of neurotransmitter inside of the synapse, and it's bound to bind to one of the receptors eventually, um, leading to those signaling cascades. Oh, sorry. Yeah, so that's like a common, like that's typically the medication that you'll see for serotonin. And so typically serotonin is created in the Ralphin nuclei, which is in the brainstem and the mucosa cells of the intestine, which honestly I feel like it's like pretty weird. Um, yeah, I don't really know how to remember, like it's just in the intestine, just like more or less that it's like a really weird thing, but I doubt you'll be asked anything like specifically about where it's created. Um, Some of the structures that the serotonin impacts is the hypothalamus. So remember I was talking about like the hypothalamus is kind of like the mob boss of the like brain or just like your neurotransmitter and hormones. So it's associated with like appetite, sleep, thermoregulation, just like bodily functions in general when you're talking about serotonin. And then also cortex, it can be associated with memory and learning. And then the brainstem, which the brainstem is like where you have your most primitive structures for the brain. So like that's like will be associated with like your sleep and wake cycle. And then we have ACH, which is the parasympathetic main neurotransmitter. And that's really important to remember. And it's also involved in voluntary. So your only real voluntary muscle is your skeletal muscle. And then muscle control. So like muscle control is like really complex. So that will not be covered in this video. It'll more or less be I'll cover it eventually. And if not, there's a ton of great videos online. And also just like great sources on Reddit too. And then too little of ACH is typically linked with Alzheimer's. And then we have epinephrine and norepinephrine. So, first I'll cover norepinephrine instead of epinephrine. So nor means parents. So, and it's typically local. So nor, like, think about it this way, right? Like if you're a parent, you're probably gonna stay like in your hometown and you want like kind of what's best for your kids, right? Like you want them to fly away, leave the nest, start their new life. And that's kind of like what epinephrine is. So just remember the nor prefix meaning parents, and it's kind of easy to remember which is which. And it's a neurotransmitter that's associated with the sympathetic nervous system. So that's also really important. So ACH, parasympathetic, norepinephrine, sympathetic nervous system. Remember, the parasympathetic is like that rest and digest, whereas sympathetic is fight or flight. And norepinephrine can also be created in the adrenal medulla as well as in between your neurons. Whereas epinephrine is your body-wide adrenaline hormone and neurotransmitter, and it is released primarily in the adrenal medulla. That is kind of all I have to say about neurotransmitters. Hopefully that video was okay and it wasn't too much of me rambling about stuff that isn't relevant. Hopefully you have a great rest of your day and this video was somewhat helpful.